Hello and welcome to your video lecture on section 10.3, where we're going to talk about the control of the cell cycle. So in humans, the frequency of our cell turnover is going to range from a few hours in early embryonic development to an average of 2 to 5 days for our epithelial cells, and to an entire human lifetime that is spent in that G0 phase by our specialized cells like our neurons and our cardiac muscle cells that we mentioned in the previous sections. And in our rapidly dividing human cells with our 24-hour cell cycle, we're going to see that the G1 phase is going to last about 9 hours, S phase lasts about 10 hours, and the G2 phase lasts about 4.5 hours. And our mitosis phase is going to last only half an hour, just to give you some perspective here. And our fertilized eggs and early embryos of our fruit flies will have a cell cycle that's completed in about 8 minutes. Again, just to give you some perspective. And so let's move into our learning objectives. We want to understand how the cell cycle is controlled by mechanisms that are both internal and external to the cell, explain how the 3 internal control checkpoints occur at the end of G1, at the G2M transition, and during metaphase, and describe the molecules that control the cell cycle through positive and negative regulation. So let's first talk about how the cell cycle is influenced by external events. The progression of our cell cycle isn't just controlled inside the cell. It's also going to be regulated by signals and conditions outside the cell. So one example is our human growth hormone, or HGH. And this hormone plays a key role in stimulating cell division. If our growth hormone levels are reduced, then cell division is going to slow down or maybe even stop altogether. On the other hand, if we get too much of our growth hormone being produced during childhood, it can lead to gigantism, where the body is going to grow excessively tall because there's too much cell division taking place. Another external factor is cell crowding. When our cells become too densely packed, they're going to send their signals to stop dividing, and this prevents uncontrolled growth and helps tissues maintain the right size and shape. All right, lastly, we have the size of the cell itself that can act as a trigger. As our cell gets larger, we've talked about in the past that its surface area to volume ratio is going to decrease, and this makes the cell less efficient at moving materials in and out. We won't be able to get the nutrients in toward the center of the cell, and the waste is going to start to pile up inside the cell, and we're not able to move it out. So to restore efficiency, our cell will divide, and that will reduce its size and improve that ratio. So along with our external signals, our cell cycle is also tightly regulated by internal checkpoints. And these checkpoints are going to act like quality control stations, making sure the cell only moves forward in the cycle when everything is ready. So the main idea is that the daughter cells must be exact duplicates of the parent cell. If we have errors take place, such as having incorrect DNA replication, then those mistakes can lead to mutations, which can be passed on to future cells. So we don't want that, and that's why we have these checkpoints that are so important. So what is a checkpoint? A checkpoint is going to be a control point in the cycle where it could be stopped. The cell cycle can be stopped, that is, until our conditions are favorable. If something goes wrong, the cell can stop, repair the problem, or if the damage is too severe, it can self-destruct through a process called apoptosis, or cell suicide. So there are 3 main checkpoints that we need to remember. The first one is near the end of G1 before DNA synthesis begins. And then we have one that's kind of at the G2 and M transition. So before that cell is really entering mitosis. And during metaphase, so this is where we have our M checkpoint here, we use this checkpoint to make sure that our chromosomes are properly aligned and attached to the spindle fibers. So let's go through each of these checkpoints more specifically. Our G1 checkpoint is going to be one of the most important control points in the cell cycle, and it'll determine whether the conditions are favorable for the cell to proceed with division. It sometimes is called restriction point in yeast because once a cell passes this checkpoint, it's going to become committed to the process of cell division. So at G1, the cell is going to check for a couple factors. Do we have adequate energy reserves? Is the cell large enough to divide properly? And most importantly, is there any genomic DNA damage that needs repair? Our external influences like growth factors also will play a big role here in helping our cell move past this checkpoint. If the cell doesn't meet all of these requirements, it's not going to be allowed to enter the S phase where DNA will be replicated. So instead, the cycle will pause, giving the cell time to repair any problems or wait for additional signals. And once the cell successfully passes this checkpoint, it's committed to division and will go forward with DNA replication and prepare for mitosis. At our G2 checkpoint, it's the cell's final quality control step before it enters into mitosis. So at this point, our cell has already duplicated its DNA during the S phase, and our G2 checkpoint is all about making sure the cell is fully prepared for division. So it's going to check if the cell size is adequate, if it has enough protein reserves to support division, and third, if all of our chromosomes have been properly replicated without any DNA damage. If errors are detected, the cell cycle will pause once again, just like the G1 checkpoint, and our cell will then attempt to either finish replicating its DNA or repair any damaged DNA before it moves forward. Only once we have satisfied all these requirements, we can have the cell safely progress into mitosis. The final checkpoint in the cell cycle is the M checkpoint, also called spindle checkpoint. And this is going to occur near the end of metaphase, as we mentioned earlier. And its main job is to make sure that every single pair of the sister chromatids are properly aligned along that metaphase plate and attached to the spindle microtubules. The spindle fibers have to anchor correctly to the kinetochore, okay, that's the yellow dots that you see there, of each of our chromatids, and they must connect to both poles of the cell. And this will ensure that when the chromatids are pulled apart in anaphase, each daughter cell is going to get the right number of chromosomes. So you can kind of see them all lined up over here after our mitotic checkpoint. If even one pair of the chromatids is not attached properly, the cell will pause at this checkpoint. That's because the next step, that separation of the sister chromatids during anaphase, is an irreversible step. Once the chromatids are pulled apart, there is no going back. So the spindle checkpoint is essential for the cell's last safeguard to prevent the chromosomes missegregation and protecting against errors that could lead to conditions like aneuploidy. Now let's talk about positive regulation of the cell cycle. Our positive regulators are molecules that actively push the cell cycle forward, ensuring that our cell is going to progress through each stage in an orderly way. And our key players are going to be the cyclins and the cyclin-dependent kinases, or CDKs. And let's talk about that cyclin protein first. So that cyclin protein is going to have levels that fluctuate throughout the cycle. They'll rise and they'll fall during specific stages, which is depicted in this figure here to the left. And our cyclins are going to bind tightly with the CDKs. And once activated, this cyclin CDK complex is going to act like a switch, phosphorylating other proteins, which you can see taking place down over here, that are necessary for moving the cell into the next phase. And note that these complexes are only going to function when cyclins are present in the right amounts. And of course, those are taking place at specific phases here. And when they're properly phosphorylated, in order to function properly. That way our cell isn't going to move forward until it's ready. There are also CDK inhibitors, and these are molecules that are going to block the full activation of CDKs. And this gives the cell a way of fine-tuning or even pausing progression if needed, and that adds an extra layer of control. So just like we have positive regulators that push the cell cycle forward, we also have negative regulators that are going to act like brakes to slow or stop the cell cycle. One key group is going to be called the retinoblastoma proteins, or RB. These tumor suppressor proteins monitor the cell size and are going to influence positive regulators. When RB is active, it's going to bind to the transcription factors. And we could see this pictured down over here. And you can see that this is going to prevent them from turning on the genes needed for cell division. Only when RB is inactivated, or inactive rather, can that cell cycle move forward. Another major regulator is going to be P53, and this is often called the guardian of the genome. This protein plays a critical role in protecting cells from DNA damage. If P53 detects DNA damage, it can pause the cell cycle and allow for repair to take place. Or if the damage is too severe, it can once again trigger apoptosis or programmed cell death. Closely linked to P53 is going to be P21. This is a protein that inhibits that CDK cyclin complex, and that directly blocks the cell cycle production. So all three of these are going to function mainly at the G1 checkpoint, making sure that our cell isn't going to divide uncontrollably. When these proteins are damaged or non-functional, cells can lose regulatory control and replicate unchecked, and this is a hallmark of cancer.